Hi everyone, welcome to Microbiology lecture series. By the way, this is Dr. R. Shyamkumar, Professor, Department of Biotechnology, Kamaraj College of Engineering Technology. So, once again I welcome you for this lecture series. So, now we are going to discuss about the topic antibiotics in microbiology. So, first let us see the contents what we are going to discuss today. What are antibiotics, classifications of antibiotics, mechanism of action, antimicrobial spectrum and different classes of antibiotics. Coming to what is an antibiotics, let us see the definition for the antibiotics. Antibiotics are derived from the microorganisms. Actually, a substance which is derived from microorganisms are produced synthetically or semi-synthetically. For example, sulfonamides, quinolones, that which kills or suppress the growth of other microorganisms. So, why antibiotics are so much of important in the field of microbiology and clinical segment? Actually, most of the pathogenic microorganisms which causes diseases to human beings as well as animals. So, to control those kind of pathogenic microorganisms, it is essential to provide antibiotics against such a pathogenic microorganism. So, antibiotics are very much important in the field of microbiology to control the pathogenic microorganisms. Then coming to the classification. The classification of antibiotics is based on various ways. Here we are classifying on the basis of mechanism of action and on the basis of spectrum of activity. So, what kind of mechanism involved to control the microorganism, that is one part, and another part is on the basis of spectrum of activity, either it is broad spectrum or narrow spectrum. So, based on that we can classify. And coming to the mechanism of action of antimicrobial agents or antibiotics. So, there are 4 major mechanisms involved to control the microorganisms, especially pathogenic microorganisms. And the first one is inhibition of cell wall synthesis. For example, penicillin, cephalosporin, bezitracin, vancomycin. And second is inhibition of function of cellular membrane. So, that may supposed to increase or decrease the permeability of the cytoplasmic membrane. That is the another action. For example, polymyxins. And the next category is inhibition of protein synthesis, example chloramphenicol, macrolides and clindamycin. And next is inhibition of nucleic acid synthesis, so example quinolones, rifampicin. So, these 4 major mechanisms involved to control the microorganisms in clinical field. So, coming to this picture, this is the mode of action. This is cell wall of the bacteria, this is cytoplasmic membrane, this is DNA and RNA nucleic acid, and this is protein synthesis. So, these 4 major components will be inhibited by the antibiotics. So, first one we are going to discuss is inhibition of cell wall synthesis. There are specifically beta-lactam antibiotics, for example, penicillin, cephalosporin, monobactam, carbamazepines and glycopeptides, vancomycin, telavancin and ramoplanin. And another category is bacteriocin. This is also coming under this category to control the microorganisms through inhibition of cell wall. So, during synthesis of cell wall or preparation of cell wall by the microorganism during growth, these antibiotics can goes and binds to the specific enzymes involved in the synthesis of cell wall, whereby it prevents the synthesis of the cell wall of a particular bacteria, whereby it the structure of the bacteria will not be constructed. So, this is one of the main action or major mode of action by the cell wall inhibitory antibiotics. And next is the antibiotic which act against or act on the cytoplasmic membrane. Say for example, polymyxins A and daptomycin. So, that which act over the lipids or proteins or some kind of the conjugate proteins present in the cytoplasmic membrane of bacteria, that due to that it increases or decreases the permeability of the cell membrane. So, due to increasing or decreasing cell permeability is cellular transaction, that may be the transport of the components inside and outside to the cells will be prevented. So, the complete elimination of the components from the cell or entry of the nutrient into the cell will be prevented. Thereby, the the growth of microorganisms will be prevented, or complete destruction of microorganisms will also happens. And next major action by the antibiotics are damage to the synthesis of nucleic acids. Actually, there are some of the examples given here: fluoroquinolones, ciprofloxacin, levofloxacin, moxifloxacin, and rifamycins, rifampin, rifabutin, rifapentin. So, these are some kind of the antibiotics which act over the nucleic acid synthesis, either over the RNA synthesis or DNA replication process. Here also some of the specific enzymes involved in the DNA replication process as well as RNA replication or duplication process. The antibiotics which can goes and binds to the specific enzymes involved in the DNA replication or RNA synthesizing process, whereby it prevents the synthesis of either RNA or DNAs. So, this is another major mechanism involved by the antibiotics to control the microorganism. And next part is protein synthesis. When you are providing the antibiotic over the microorganism, what it does, it directly goes and binds to a protein synthesizing part, either it may be the enzymes or the subunits, ribosome subunits, either 50S or 30S RNA ribosomal subunits, whereby it prevents the binding of subunits during the synthesizing process in over the mRNA. So, there are some of the examples. So, that can binds with the 30S subunits, aminoglycosides, streptomycin, gentamicin, tobramycin, and some of the tetracyclines, tetracycline, doxycycline, minocycline, and some kind of antibiotics which can binds to the 50S subunits, macrolides example, erythromycin, azithromycin, clarithromycin, chloramphenicol, oxaloidones, and lincosamidins. So, these are all some of the antibiotics which can prevents the protein synthesis. So, where it can binds to the either 15S subunit or 30S subunits, or sometimes it can binds to the mRNAs, whereby it can prevents. So, these four major mechanism involved in controlling microorganisms. So, let us coming to the spectrum of antibiotics. So, we can categorize the antibiotics as a broad spectrum and narrow spectrum based on the activity or based on the action over the microorganism. Coming to the narrow spectrum, the drug that can only act on one specific kind of microorganisms or one specific group of microorganisms or one strain of bacteria, say for example, isoniazid. So, it can supposed to control over the gram positive bacteria, which will not act against gram negative bacteria. That is why this is coming under narrow spectrum antibiotics. At the same time, broad spectrum antibiotics, see the drug that can that have wide antimicrobial scope, say for example, tetracycline and chloramphenicol. So, they can act against either gram positive and gram negative organisms. So, this is what is the category based on the antimicrobial activity. On the basis of mode of action, we can categorize the antibiotics as bacteriostatic and bactericidal. You know what is bacteriostatic and bactericidal? Bacteriostatic means when you give the antibiotic over the microorganism, what it does? It just suppress the growth, it can just stop the growth of microorganism. So, after reducing the concentration in the body, automatically the microorganism again starts to grow. So, it just controls the microorganism, that is why that is static antibiotic, bacteriostatic, it will not completely kills the microorganism. So, for example, tetracycline, chloramphenicol, erythromycin, lincomycin. And another category, bactericidal. Bactericidal means cidal. What do you mean by cidal? Cidal means complete killing. So, the antibiotic which can completely destroys the microorganism, that is bactericidal antibiotic. Say for example, cephalosporin, penicillin, erythromycin, aminoglycoside, cotrimoxazole. Say for this cephalosporin, penicillin, these kind of antibiotics which can destroy the cell wall of the bacteria, whereby the complete structure of the microorganisms will be destroyed. So, ultimately the organisms will be killed, further there is no growth. That is why they are all coming under bactericidal. And on the basis of the spectrum of activity of the antibiotics over the microorganism, say here we have given some of the examples, mycobacteria, gram negative bacteria, gram positive bacteria, chlamydia and rickettsia. These are the 4 major group of organisms which can cause us life threat to the human beings in the clinical as well as the medical field. Say for example, tobramycin, it is active against mycobacteria and gram negative bacteria. At the same time, streptomycin also coming under the same category, it can control the mycobacteria and gram negative type of organisms. But in case of isoniazid, it can control only mycobacteria. So, that is coming under narrow spectrum. And here penicillins, so they can control both gram negative bacteria, gram positive as well as chlamydias. Sulfonamides and cephalosporins, they can control both the gram negative and the gram positive. At the same time, you see the tetracyclines. So, they are very broad spectrum of antibiotics, they can control gram negative, gram positive, chlamydias as well as rickettsia. And you see the polymyxins, they can control only the gram negative type of bacteria. So, this is what is some of the antibiotics activity over the microorganisms as broad spectrum and also narrow spectrum. And coming to this classes of different classes of antibiotics, the first thing is this is based on the functional group and activity over the microorganisms, which can be categorized beta lactams, aminoglycosides, glycopeptides, anazomycins, quinolones, streptogramins, lipopeptides, sulfonamides, chloramphenicals, tetracyclines, macrolides and oxolidinones. So, coming to the beta lactam, it inhibits the bacterial cell wall biosynthesis, say for example amoxicillin, flucloxacillin, and coming to the aminoglycosides, they can inhibit the synthesis of protein, example streptomycin, neomycin, and glycopeptides. This is the conjugative peptide associated with the glycan molecule, inhibits bacterial cell wall biosynthesis, example vancomycin and teicoplanin. Anosomycins that inhibits the synthesis of RNA by bacteria, that is geldamycin, rifamycin, and quinolones. It is another category of antibiotic which interfere with the bacterial DNA replication. Example ciprofloxacin, levofloxacin and streptogramins. It inhibits the synthesis of proteins, example prestonomycin 2A and 1A. The lipopeptides, it is another group, disturb the multiple cell membrane function. It is active over the cytoplasmic membrane of the bacteria, example daptomycin, surfactomycin and sulfonylamides. It prevents the growth of bacterial multiplication, example prontosil, sulfonylamide. Then chloramphenicol, it is another type of antibiotic which can controls or inhibits the protein synthesis of the microorganism. And next is tetracyclines, it inhibits the synthesis of protein by bacteria. Tetracycline, doxycycline and oxytetracycline. The next category is macrolides, they inhibits the protein synthesis of bacteria. Erythromycin, clarithromycin and azithromycin. And next last one is oxolidinones, that inhibits the protein synthesis of the bacteria, example linezolid and cycloserine. So, these are all some of the example and classes of antibiotics. So, till now we have seen about the types of antibiotics, mechanism of action and broad spectrum, narrow spectrum, and finally the classes of antibiotics. So, thank you very much for your attention. Thank you.